Hi everyone, welcome back to Cells to Cell. My name is Heem, that's Nick, and we have a very special guest with us here today. This is Ryan Hall. You want to introduce yourself?
Yeah, hi guys, I'm Ryan. I was born and raised here in Cleveland, so I'm a local. I did my undergraduate education here at Case Western Reserve, and I'm here doing a Ph.D. as well, currently in my 5th year in the Biomedical Engineering department. So.
That's awesome. Heem, what are we going to talk about today?
So today we're actually going to be talking about nanoparticles. Ryan works in a lab that has developed its own nanoparticle, and we're going to get to know more about that. And they use this nanoparticle to send gene therapy agents to cancers and specifically.
Yeah, Ryan, if you could, for those watching who may not know, can you kind of just give like your basic definition of gene therapy? Like what is it? What does it do? What are we trying to do with it? That kind of stuff.
Well, at least in our lab, we work with something called RNA interference, where we're essentially trying to affect the expression of specific genes that are expressed in cancer to have some sort of a desired effect. So in our case, we try to target genes that are related to the epithelial to mesenchymal transition. So basically a process where the cancer goes from growing in place to spreading throughout the body. So we're trying to essentially prevent that from happening. And we use RNA interference and gene therapy to do that.
Yeah, preventing metastasis is really important because that's when you get into a lot of trouble. So, Heem, you actually worked in this lab last summer, actually. That's how we got this contact. So share maybe just a little bit of your experience of, not even necessarily what you did in the lab, but kind of what's an atmosphere of a lab like? Because that's something we haven't talked about on this podcast yet, and I think that some people might be wondering what it's like.
So I was actually trying to find a place to work with over the summer to get some experience, and I took nanomedicine as a class here at Case, where Dr. Lu, the PI of the lab Ryan's working in, he taught the class, and he was telling us all about, he would teach us about the nanomedicine industry, and then he would just throw in there casually, I've developed my own nanoparticle, and it just so casually, everyone was like, okay, yeah, he developed his own nanoparticle, but then to me, I was like, holy crap, that is so cool, I want to learn more about it. And then I emailed him, and I was like, hey, is there anyone I could work with? And he partnered me with Ryan.
That's awesome. And
then I met Ryan, he gave me the whole rundown on everything he's doing, and I was so interested in everything because he's doing something so relevant too, because squamous cell carcinoma is what he works on, and it's a cancer that's highly prevalent in a lot of people, especially in the dental industry. The other day I was talking to a dentist, and he was like, yeah, I have patients who come in with squamous cell carcinoma, and I was like, oh, that's funny, I know somebody that's working on that, and I really enjoyed it because the lab does such relevant stuff, for cancer especially, and learning about the past members of the lab who Ryan knows and everything super well, the work they did in terms of chemistry and stuff, and then his biomedical engineering background and everything, and how everyone has to work in a team together, and every Monday they have lab meetings where it's just you just have to hear what everybody else is doing, so kind of what the lab is all about and everything, and I think that was a really good atmosphere. And every day Ryan always helped me so much. He gave me after the first day, I came in, he just put a pad on my on my desk with a pen, and he's like, you're gonna take notes.
Wow.
So on every procedure I took all the notes. I got to know everything, every detail about all the machines, how they work. And he's actually in charge of a lot of the stuff that goes on in the lab, the -80 freezer and everything that they had problems with when I first came in, and that was a lot of struggle. So it's a lot of on the feet moving and on the feet thinking and knowing how to problem solve.
Yeah, I think that's important because when people think of labs, they think of, I took a biology lab in undergrad. It can't be, it can't be any different, where the environment and what you're doing is, incredibly complex compared to just looking at a microscope. So can you kind of speak on that? How would you say, just kind of what's your daily workflow? You punch in at the lab. What do you kind of do without going into too much
detail?
Right.
It really just depends on the time of year, what stage of your project you're working on. and what particular experiments you have going on. But I mean, it's almost kind of like a 9 to 5 most days, with the exception of a handful of really long days and maybe some days where it's not quite as busy.
Yeah.
But there are experiments that take all day, 6, 7, 8, 9 hours.
Yeah. I remember cell biology. We would have it's a 3-hour lab and we'd be there for 4.5 hours, and that's just for an experiment that we have planned out and we know what should happen. I can't. I've yet to do any of like personal research. Just along the lines of like in a lab with my own cell culture, cell line, working with whatever it is, nanoparticles, and it seems like the transition would be really hard for someone going from maybe like an undergrad or a master's to all of a sudden like a PhD. And I know that a lot of people maybe listening to this might be interested in PhDs or already have their PhD. I know that I've thought about a PhD, so kind of what was your process, from undergrad? Did you go straight into your PhD? Kind of how did that process go? Because I hear horror stories about PhDs, and I hear horror stories about, finding an advisor and picking a project, and you get, so close to finishing your project, and it just doesn't work, so you don't have a thesis or for your dissertation or anything. So, kind of elaborate on that too, just like, general quality of life of a PhD.
Yeah, so I pretty much went straight into graduate school right after undergrad. The lab that I'm currently working in for my PhD, I was an undergraduate working in that lab as well, so it was pretty easy for me to pick out the PI or the professor that I was going to be working under.
Yeah.
But transitioning from being an undergraduate doing research to being a PhD candidate doing research is a pretty stark difference. As an undergraduate, you're typically working under another graduate student or something, and you're being told what to do or what experiments are going on. You're not the person really like keeping the project together per se. You're just contributing to it. Whereas once you become that PhD candidate and you begin working on your own project, it's like that's your project. You are in charge of everything, and you have to do everything within that project and sort of take full responsibility for that. And one of the things that I've learned through the PhD program is you have to learn how to troubleshoot and problem solve. There are countless times where experiments might not work, or the theory behind a project might not pan out quite as you thought, and some of it could just be experimental error, or some of it just could be you're not quite treating the cells in just the right way you're supposed to be treating them. So a lot of PhD work, in my experience, is kind of figuring out what the problem is and learning or trying to figure out how to solve that problem and the procedures of going about that.
Him working under a PhD, can you attest to that?
Yeah, I mean, Ryan was always problem solving. If there's not enough reagents, he would be in charge for ordering them. When I came in, like, the floor above had some water issue where like it leaked down onto our lab area. So there's so much that went wrong.
That's still getting solved.
That's still getting solved, yeah. So he had to put in a bunch of like insurance claims and whatnot for it. But I think Ryan actually has a really unique experience and perspective because he's seen how the lab has evolved. He's gone through so many different people coming in and out of the lab, and he's even seen where like, for example, the ECO nanoparticle, that's what Dr. Lu has named his nanoparticle ECO. We'll go over that. But Ryan, from an undergrad, has seen how the problems back then is like not being able to mass produce the ECO nanoparticle to having, what was his name?
Andrew.
Yeah. Andrew figuring that out, and you seeing his process, and then Nadia and everyone, and then now you being that guy, and you see Hong under you and everyone just learning from you. So how is that? Can you just go over like where was the lab, and where was like the progress of the ECO nanoparticle when you started to where it is now?
Yeah, the lab definitely it changes a lot over time. Give it 4 years and the basic underworkings of the lab itself will change, and that's no more stark of a difference than with the ECO. When I came into the lab, the nanoparticle that we work with, it it was very difficult to mass produce, to purify out, to make sure that we actually had a large enough quantity to do like in vivo studies and mouse work. And one of our previous graduate students, as he mentioned, Andrew, he spent a couple of years of his Ph.D. actually doing just basically chemistry all day, every day of the week, trying to solve and figure out how can we produce more of this, ECO. And sure enough, he figures that out, and next thing you know, we have ECO for days. Like we'll never essentially have issues with ECO ever again. We're able to produce more than enough for what we need to do with it, that sort of facilitated a lot of our projects, because when you're struggling to produce these very important resources for the lab, once you sort of have enough of that or an abundance of it, all of your other projects that you're trying to use that for can just sort of like come forth and be completed, or at least be worked on.
I think that's so interesting, because years ago, so like 6 years ago, the lab was in a state where it's just like they were trying to figure out how to make this nanoparticle so it's usable by everybody else in the lab. So like everyone was working to figure that out, especially Andrew. So what was everyone else in the lab responsible for while ECO was not being so used by everybody?
Yeah. So my lab itself actually has a few sort of working components. We have like a chemistry department, like that's where Andrew worked, and we've also had a few other students come through working on some other projects in that regard. And we also have at least probably half of our research is focused on using magnetic resonance imaging for targeting cancer. And so while the ECO issue was getting figured out, a lot of our projects ended up actually focusing on the MRI in the meantime. Okay. And at least with everything that's been done so far, we use a targeted MRI contrast agent that was actually developed in the lab. It's targeted with a little short chain of amino acids, a little short peptide, and it selectively targets cancer. So we've sort of done a handful of preclinical studies using various different cancer types, like head and neck cancer, what I work on, breast cancer, prostate cancer, and lung cancer as well, and pancreatic cancer, can't forget that. Yeah. And we show that this targeted contrast agent that we've developed is actually in many cases more efficient at producing contrast enhancement than clinical agents currently being used.
That's amazing.
That's awesome.
That's so relevant.
Yeah, and it sounds like once you guys got like the CEO or ECO.
ECO,
yeah. Not the CEO.
It's all the acronyms.
Once you guys got the ECO, mass produced where everybody could use it in the lab, it just sounds like kind of that downhill effect. Once it's available, everything just kind of starts falling in place to get you to the point where you guys are now. So, just quickly, when did that happen? When did the ECO become available for everybody to study with, to study in the lab? And where are you guys now? Was it last summer when he was there? Was it the summer before? Was it the winter before? Kind of just a little timeline.
Yeah, it was maybe 3 years ago now or so. It was maybe a year, somewhere between a year to a year and a half after I had joined the lab. And yeah, once that happened, it was just a windfall of projects coming off of that.
That's awesome.
Yeah,
that's awesome. I feel like a lot of labs get kind of caught up in, they can't get that one reagent that they really need, or their cell line just won't cooperate, and you're troubleshooting that while you're also trying to continue your research, and it's while biology is awesome and studying this kind of stuff is a lot of fun, it comes with its roadblocks. Like no study's ever going to go perfectly and get you perfect pretty results to be posted in a paper, or to be put into a product. It's important to understand the scientific method comes with a lot of roadblocks.
I was just going to say, that's especially when you're producing a therapy or or a lipid or some sort of molecule for medical use, that's one of the things that you really have to make sure you get right, is actually being able to upscale production.
Yeah.
Because if it's practically impossible to produce efficiently, then
there's no point.
There's really very little point.
Yeah, speaking on, kind of the end goal of these, nanoparticles, on this podcast we cover, a little bit about like the FDA and how they view like cell therapies. So how does that differ with these nanoparticles? Are they looked at as, just like your like original type of medicine where, you go through the normal, phases and trials, or are they kind of like in their own area, like the mesenchymal stem cells, for example?
I feel like nanoparticles, and especially with things like gene therapy, is definitely sort of like its own little area. You didn't really see much in the way of gene therapies until really the COVID vaccines came around.
Yeah. The mRNA vaccines were a giant leap in science, and I don't think a lot of people really understand that. Going away from the classic viral vectors and attenuated viruses, all of a sudden to this new novel little piece of RNA that your body can just read and make antibodies against.
Yeah, and now that those have been produced as well and they're being clinically used, I mean, the gene therapy field as a whole has seen a massive boom.
Yeah.
Because practically everybody's trying it now.
Yeah.
So why don't we break down like what the point of a nanoparticle is? So like, why did Dr. Lu think doing nanomedicine was important? So like, what's the point of it, and why is it used?
Yeah. So I'll talk about it in our sort of situation here. So because we work with things like small RNAs, there's a lot of issues that come into play if you were to just say like inject an RNA into your body. The primary thing would just be degradation of the RNA. So if you have this therapeutic that you're trying to deliver and it actually degrades in your blood before it can even get to where it's supposed to go, there's really no point in even trying to treat with that. And another thing too is that most therapies these days are not like targeted specifically to things like cancer cells or to whatever target cell you're trying to get it to. And in a lot of cases, if you were to try to target a specific like chemotherapeutic or specific RNA, you might end up actually affecting the function of that therapeutic or that RNA that you're trying to deliver. So what we try to do with developing nanoparticles is provide a system where you prevent degradation of your cargo, your therapeutic, while also providing a system where you can put things like targeting ligands onto the outside of the particle without affecting the actual cargo of your actual therapeutic itself.
Yeah, I think that's important, especially it reminds me going back to my medicinal chemistry course in undergraduate, when we were talking about pharmacodynamics and pharmacokinetics. You're worried about, the absorption, the distribution, the metabolism, and the excretion. And that's important, especially when you're looking at drugs, and especially when you're looking at, I'm assuming, are these nanoparticles put in like micelles, or like, little, bilayers to carry something on the inside while having those peptides on the outside that are like kind of targeting the cells. But, like you said, if it gets into your bloodstream, it just immediately gets broken down. There's absolutely no point. So on that topic, how do you guys administer these nanoparticles? Is it, I'm assuming it's going to be IV, but would there ever eventually be a possibility where people could be taking nanoparticle medications at home, like through pills or something?
Oh yeah, so in our case, we've only ever really worked with intravenous injections. At least currently, it's kind of like the most reasonable route to use, just because you get essentially 100% bioavailability in the blood through that IV injection. But obviously, with things like patient compliance or having to go to get an actual like procedure done, get an IV done to receive the therapy itself, things like pills or things like nasal sprays, stuff like that, really sort of they're sort of like a topic that is really worth looking into.
Yeah.
You mentioned that you asked if like these nanoparticles are put in my cells. Not quite. They are their own little lipid. Okay. So do you want to break down E, C, and O?
Oh yeah. Yeah, so
I guess
so we've been talking about it for a little bit now.
Yeah.
ECO, it's the lipid that my professor in the lab developed. It's got 3 specific components. The E itself stands for ethylene diamine. All that really means is that it's got 2 nitrogens on it, and that's important because nitrogens can actually take on a positive charge.
Right.
And then it's got 2 cysteine molecules. That's what the C stands for. The useful thing about cysteine is that it has a free thiol group. So we've got 2 free thiol groups, sulfurs, on our particle, and that allows us to actually attach things to it. And then in true lipid fashion, we have to have a lipid tail. So we have 2 oleic acids. That's the O. So ECO, ethylene diamine, cysteine, oleic acid.
And now the E is going to be on the inside, and since it takes on a positive charge, you can actually put your negatively charged RNA groups and also DNA. Can you put DNA?
Yeah. So we've worked with small RNAs. We've also worked with DNA plasmids as well. One of the members of our lab has actually worked with eye diseases like Stargardt disease and Leber's congenital amaurosis. And in both circumstances, he used a DNA plasmid and it worked beautifully awesome. With these ECO nanoparticles.
Yeah, it sounds like there's we're on the verge of a bunch of like customizability with these things.
Oh yeah.
Which, we've been, as humans and as researchers, we've always been looking for, the silver bullet, or at least some type of personalized medicine that can be used in a bunch of different people for a bunch of different stuff. And it sounds like that this is a pretty good path that we're on.
So one more thing.
Yeah.
Some nanoparticles are actually also developed with metals like iron oxide. Some people are trying to develop gold nanoparticles. So in this case, it's a lipid because they degrade into things that your body can process. If you have something like gold or any other metal, they really can't filter through your kidney, right? So why choose the lipid over something else for a nanoparticle?
Well, in our case, at least, with working with RNA or DNA plasmids, stuff like that, there's a lot of negatively charged phosphates on the backbone. So working with a lipid or some molecule that exhibits a positive charge, like the ethylene diamine head group of ECO, you get that natural automatic interaction between that positively charged nitrogens and those negatively charged phosphates, and it sort of facilitates sort of like automatic generation of particles once you mix the 2 of them together. Whereas working with something like gold nanoparticles or iron oxides, I haven't personally done a ton of research into that, so I can't say for certain.
Yeah.
But there might be a little bit more chemistry involved or some other thing like that.
And now the lipids on the outside make sure they do some electron shielding to make sure that everything else throughout circulation does not cause any sort of toxicity or doesn't even react with your inside agents. Is that right?
Yeah, well, so the lipid itself, once you form that nanoparticle, you do get shielding of your cargo, your RNA from whatever is in systemic circulation. So any enzyme that'll degrade RNAs or DNAs in circulation, your cargo itself is going to be protected from that. Now, the actual charge shielding of the nanoparticles themselves comes into play when you look at things like pegylation, so attaching like polyethylene glycol or targeting ligands to your particle. And once you do that, you kind of have these long chains of these PEG polymers attached to the outside, and it'll sort of shield your particle from all of these other like proteins and things that'll interact with a positively charged molecule in the blood.
Nice.
Yeah, that's awesome. So it's probably one of my last questions, and it's a big one, but how close is this to being a finished product? How far away can we see this? From starting in phase 1, maybe clinical trials where you're testing safety on human patients.
Yeah, well, that's kind of a tough question. There's a lot that you have to do before even getting into that phase 1. There's a lot of preclinical work that goes into it, and also a lot of money that has to go into getting clinical trials started. But we're working our way through getting studies done with specific treatments with this ECO nanoparticle, and assuming that all of our studies come out with positive results, you might be able to see down the road, a clinical trial that uses ECO or something similar to it. I can't say for certain how long that'll be. It's kind of hard to say, but there are certain treatments, like the treatments for the eye diseases like Stargardt's disease, has received a little bit of press. Through Case Western's own news outlets as well. Dr. Lu's research with ECO it's not a completely unknown. It's definitely getting some attention. And thus far, it seems to have almost universally positive results.
Awesome. That's awesome. The reason I asked that is because, again, I think sometimes people forget that when we get a new medication on the market, the research started 8, 10, 12, 15 years ago.
Oh, yeah.
So, joining this lab and you guys, just finally getting the ECO to be mass produced to where you can do, your studies with it on all these different types of cancers, which is wonderful, by the way. It's incredible, what this lab is doing. But, people have to understand that it takes time and it takes regulation and safety and everything like that. And, it's nice to have someone come on the podcast who is actively in that field and actively working to do the studies, get the numbers, and, kind of push it to the next level. And I think that's commendable, and it's really cool and, inspiring for me, for him, and for, maybe some people listening. I think it's really cool.
Yeah, and, gene therapy itself, whether you're using plasmids to get expression of some specific gene or using RNA interference like I do, to try to affect the expression of genes that are naturally expressed, it has so many applications. And you look at things like the mRNA vaccines. Once those get approved, that just opens the door to trying to use the same platform to treating all sorts of conditions and diseases using things like gene therapy. So with the ECO nanoparticle, once we get the first breakthrough and assuming it goes to something like a clinical trial and exhibits success there, it kind of opens the door to treating so many other diseases.
Yeah.
And it's so much more versatile than like a standard chemotherapeutic.
Right.
And so in this podcast, we're actually going to include 2 papers. One is for the ECO and one is for the ZD2 contrast agent. So now both of those 2 things combined, let's go over kind of what you would practically see in the lab. So if a mouse has cancer, you first give them the contrast agent and you image them to first see where the cancer is, right? Then you give them your nanoparticles with this RNA, and then you let it circulate, so then it goes into the cancer cells and does its effect. And now, do you put in the contrast agent again after? So every time you dose them, do you put another contrast agent? Is that how it goes?
Yes, we do. So, taking those 2 things and combining them is actually quite a unique thing, I think. So with cancer and our targeted contrast agent, we're trying to visualize the expression of a specific form of a protein called fibronectin. It tends to be very overexpressed in many cancers. And so using this targeted contrast agent, we actually get really, really good visualization of the tumor itself. And what's nifty about that is the treatment that we're using, and this has been published with breast cancer as well. The treatment that we're using is a microRNA that targets fibronectin. So when this microRNA goes and targets fibronectin, it's actually reducing its expression. So when you do deliver your contrast agent and perform MRI before your treatment, and then you provide your treatment and then you do MRI again with that same contrast agent, you can see the change in fibronectin expression as your treatment is progressing.
Wow,
that's awesome.
And
so it's like
it kind of gives a readout of how successful your treatment is.
So your gene therapy and your contrast agent target the same fibronectin expression, and in theory, your gene therapy reduces the expression of fibronectin, and in result, you should have less uptake of your contrast agent, so you'll see less of it. And now, why do you use MRI? Because people like to use CT and all these things, and X-ray. So MRI is a very unique thing also that I feel like we should touch on.
So we use MRI in part because it provides such excellent soft tissue contrast. With a CT, you might not be able to visualize the different organs in the body, or sort of at least easily see the difference in tissues in the abdomen or wherever the tumor is. Whereas with a modality like MRI, you can identify specific organs, you can identify the tumor itself, and you can see it with really quite good resolution.
And radiation is the biggest thing.
Oh,
too.
With obviously with CT, you do get ionizing radiation. If you were to use something along the lines of PET imaging, positron emission tomography, the contrast agent itself, the radioactive agent, is radioactive. So you could run into issues there. Whereas with MRI, you don't have any ionizing radiation, and at least thus far, there's unless you have like severe kidney diseases, there's really no known complications for gadolinium agents aside from in patients that have severe kidney failure.
So now imagine if you're trying to image a cancer patient, you give them radiation through CT or X-ray, and then you mutate that same cancer because you're radiating it. Whereas the approach that the Lu Lab is doing is so smart, and it's so unique, and it's so applicable and relevant to what needs to be seen in the industry, because not only do you have MRI, you can do so many things with it, and it's highly targeted with the nanoparticles, and I think it's really awesome.
Yeah.
Really awesome to see what the Lu Lab's doing.
I just can't get over the specificity of nanoparticles. I think that it's so cool how you can just target, specific cells, and especially specific cancers. And I did have a question about maybe another, possibility of this type of medicine. So, right now you're working with RNA interference or RNA interferons. Would there be any possibility that you could ever, add a gene to be expressed in a cancer cell? Or, I mean, I'm sure it's probably been done. But, a gene that triggers, the apoptosis or, cellular death pathway.
Yeah. Now that last one would be a little tough because you would have to make sure that your therapy is so extraordinarily cancer specific.
Yeah.
But really, that's where things like DNA plasmids come into play, and the lab member working with eye diseases like Stargardt disease, and they have a deficiency in the expression of a specific gene. I personally don't know the gene itself.
It's okay.
But we deliver this plasmid, and we actually get those retinal cells to start producing that particular gene and protein again. Yeah. So you can definitely apply, this technology for getting your cells, whatever target cells you're trying to look at, to express a specific thing.
Yeah, that kind of reminds me of how we're trying to treat cystic fibrosis right now. So cystic fibrosis in the lungs. There's a chloride deficiency, which makes it all kind of mucousy and like pussy. And researchers are actually trying to find ways to inject. And I think some people have actually used, they've tried DNA plasmids through inhalers, where you inhale it in and it's trying to get picked up by all the bronchi. And, even down in the alveoli level. And, get there and, start expressing that gene to fix it. The problem is a chlorine transporter in the membrane, and I think that's really cool. And it, that kind of made me think, how maybe we could do nanoparticles through. When I asked about intravenous injection, other ways that it might be possible, because there are plenty of ways to to give new novel drugs, especially with the eye disease. Like, what if they created eye drops that was easier to just get absorbed in the eye, and there's less chance of it going through the blood and getting caught somewhere else in the body, and you can get like a pretty good delivery rate. I don't know the numbers of what they have. Maybe they have 100% already, but maybe not. And maybe that's kind of one way to kind of turn, but that would be a whole nother step in the process, which I get. It would be a lot of money, but it'd be cool. I think it'd be cool.
Yeah, and the different routes of delivery too, they open up new opportunities. Like if you were able to like aerosolize, your treatment in some way to treat, lung diseases or lung issues, versus, doing something like eye drops. Currently, we're working on just injecting straight into the eye. So obviously that's a whole procedure there, but toying with the different routes of delivery, and if you find something that works for nanoparticles in multiple different ways, it just expands that versatility even more.
And that's what we're looking for, is versatile medicine.
Most people, when they think of regenerative medicine, will usually think of stem cells, but if you can use gene therapy and nanoparticles to restore somebody's eye function or take away their cancer, that is regenerative medicine in a nutshell. So
absolutely.
I think including nanoparticles and gene therapy is a huge part of regenerative medicine that needs more attention, because right now stem cells are like the golden child, it's just like stem cells will do everything, but there's a dark side to the stem cells too that I feel like nanoparticles and gene therapy can definitely make up for.
Yeah, that's awesome. Well, we appreciate your time.
Absolutely.
Thank you, Ryan.
Thanks for having me on.
Yeah,
our first
guest. First guest. And I don't think it'll be the last time you'll be on here either. That was a great session. And I think that I definitely learned a lot, because, like I said, I have not taken nanomedicine. I know very little about nanoparticles, and now I feel a little more confident when I go through and read papers about them and everything. I'll have a little bit more of a basis. So, Hemi, got anything else to say before we sign off?
No, that's all from me. And do you have anything else to say?
I don't have anything else other than thank you guys for having me on, and I'd be happy to come on again.
It was a pleasure. It really was. So, all right. Well, I'm Nick. That's Hemi. And
I'm
this
Ryan.
and this was Cells to Sell. Thank you, guys.
See ya.